Welcome everybody, my name is Juami van Gils and today I would like to tell you something about the effect of disordered flanks on the aggregation of amyloid fibrils in neurodegenerative disease. Amyloid fibrils are protein aggregates that aggregate very specifically into a beta-stranded form, as you can see here on the right. And typically proteins that aggregate into amyloid fibrils have a relatively high level of disorder and some propensity to form these beta strands. Now, these amyloid fibrils are found in many different brain diseases, such as Alzheimer's, Parkinson, Huntington, and they are thought to be associated with the disease. So in previous work, we have looked at the aggregating part of these amyloid fibrils, highlighted in purple, and we found that hydrophobicity is very important to drive the aggregation of these fibrils. So the next question we had is, okay, so then what is the role of these disordered flanks? Which leads us to our research question, which is what is the effect of disordered flanks on amyloid fibril growth? Now, to study this question, we use our model protein, which is alpha-synuclein. This is the protein that's associated with Parkinson's disease. And here on the left, you can see monomers in their functional state as they're bound to a micelle. And on the right, you can see the amyloid fibril. And what you can see, so here in the middle, you see the aggregating core. Then on the left, you see the N-terminus, which is slightly positively charged, is highlighted in red. And then the C-terminal disordered flanks are very negatively charged, as shown in blue. Now, to study the growth of these fibrils, we use a technique called quartz crystal microbalance. And basically what you do is that you have a gold chip with your amyloid fibrils attached, and you stimulate that with an electric current, and that will cause the chip to vibrate. And so what we can measure is the frequency of this vibration. Now, if you add more monomer to the chip, and this leads to amyloid fibril growth, then the frequency of the vibration will go down. So in this case, very important to realize that if we see a decrease in the frequency, this means an increase in mass on the chip. Now, our setup is actually quite simple. So we have a chip with our alpha-synuclein fibers attached. Then in the one situation, we leave the fibrils intact, and in the other, we degrade the disordered flanks of the fibril. Now, in both cases, we add new monomer to the chip, and then we see what happens to the growth of alpha-synuclein. So first, the results where we leave the fibrils intact. So here is, on the x-axis, we see time, and on the y-axis, on the top, we see the change in frequency of the vibration, and at the bottom, we see the slope of the graph on the top. So here we see, for example, in all three of the growth phases highlighted in purple, that if alpha-synuclein is allowed to grow, the frequency goes down, which is what we expect. And what you also see is that in all three of these phases, the growth rate is approximately equal. So it doesn't change, which makes sense because we didn't do anything to the fibril. Now, if we, in between these steps, use proteinase K to degrade the flanks, then suddenly we see that the rate of growth increases after every degradation step, which means that in addition to linear growth of the fibril, something extra is happening, which makes it grow faster. Now to explain what's really happening, because of course here we're only measuring the frequency, we developed mathematical models that could give us some mechanistic insight. So we have a null model, which is this growth of intact fibril. Then model A is where if we degrade the flanks, the fibril still just grows linearly. Model B, the fibril will continue to grow linearly, but also monomers can bind to the surface where the flanks are no longer intact. Model C does the same as model B, so monomers can also bind to the surface, but this leads to the growth of an additional fibril on top of the original fibril. Now in model D, again the same as model B, but here, binding to the surface leads to branch growth of fibrils. Now, as you can see on the right, so model C and D are here indicated in red and purple, and they lead to the fastest increase in growth rate and correspond the best to what we've seen in our experimental work. So this suggests that either model C or D is happening. Now, if the growth if the surface binding of these monomers to the fibril leads to the growth of new fibrils, we call this secondary nucleation. And so in conclusion, we can conclude that the disordered flanks slow down the amyloid fibril growth, And the mathematical model suggests that the increase in growth that we see when there's no flanks present is due to secondary nucleation. And with that, I would like to thank everybody who's involved in this project, both on our side here at the VU and our collaborators in Denmark, and of course our funding. I'm open to answer any questions you might have about this project and hopefully see you at the conference.